Okay, so metaphase is the second phase in mitosis. So you get to see the arrangement like this in the middle. So very easy to identify metaphase, M for middle. Okay, so let's look at the behavior, the chromosomal behavior, and also other structures. First, centrioles. We talk about these centrioles. They are now at the opposite poles of the cell, opposite one another. Right, just now? Before that, they are still not opposite yet. They are migrating to the opposite side, now already at the opposite poles. So the spindle fibers maintain the chromosomes at the equatorial plane, okay, or the metaphase plate. This is known as the equatorial plane in the middle. Okay, sometimes we also call it as metaphase plate. So next is the chromosomal behavior. You say what happened to the chromosomes? They are now aligned in a single row on the equatorial plane. So they are queuing up like this with the centromere attaching to the equatorial plane, right? The sisters, this is the sister to this. Okay, the sister one another, they are aligning nicely at the middle. So metaphase ends when the centromere begins to divide. So now 1, 2, 3, 4. The centromere is still not dividing yet. You can still count 4 centromere over here, and metaphase will end when this centromere starts to divide. Okay, so this is under the microscope, you can see pictures like this. Okay, this is schematic diagram. Okay, moving on. Anaphase. PMAT, remember PMAT. Okay, so now, this is now the first two phase. You draw oval shape of the cell. But now you start to draw something like this. Okay, there's a dented region in the middle over here. So what happened is that the centromere divides. Okay, just now it begins to divide. Now it divides into 2 already. So you can count 1, 2, 3, 4. Another side 1, 2, 3, 4. So they are already divided. So the sister chromatids already separated from each other. So please notice how to draw where the centromere is the leading one. Okay, because the spindle fiber shortens. Okay, you know that this centromere is attached to the spindle fiber, right? So when it shortens like this, pull, so this will go first. So because common mistake is that students draw the legs. You don't call this the legs lah, but just to make you understand that students draw it terbalik. This over here, they draw like this. They draw like this, which is wrong lah. Okay, the leading one is the centromere. Okay, and the legs are following behind. Okay, so spindle fiber shortens, they contract, and the sister chromatids are attracted. They're being pulled to the opposite pole. So it ends, this phase ends when the chromatid arrives at the pole of the cell. So when they are at the pole, we say this phase ends. Okay, moving on to the last phase, that is telophase. Okay, now more dented, this region. Can you see that? It's just not a bit, now very ramping already, the pinggang. So when the chromatids are at the opposite poles, now we call them as daughter chromosome. Okay, please remember the terms daughter chromosome. Daughter chromosome. Because when you describe the process of mitosis, you need to make use of the terms given. Okay, the correct terms. So now each pole, you see one, this pole, and this pole over here, each pole contains one set of complete and identical chromosomes, complete and same chromosomes. So the chromosomes are shaped again as fine chromatin threads. So that means now they revert back to the initially when they were in the prophase. When they were in prophase, remember the term chromatin? So that's why you get to see like it's a thread-like like this. Okay, so the nucleoli are formed again. Okay, some books they show the nucleoli being formed again. The spindle fibers no longer are there. They will disappear. Okay, and new nuclear membrane is supposed to be nuclear instead of nucleus. So new nuclear membrane is being formed and the telophase stage is followed by cytokinesis. Okay, remember in M phase you have, what do you have just now in M phase? You have mitosis and cytokinesis. So what is cytokinesis? It is the division of the cytoplasm, right? So the telophase stage is followed by cytokinesis. So it will end up, this will cut into two. So that's why we say you will get two daughter cells. Okay, daughter cells, daughter chromosome. Right. So, okay, maybe we can stop here.